in the next few slides, and when you look at the scope, you'll see that even though these are both cnidia, the structure of penicillium is a lot different than the structure of Aspergillus. Okay? There's also sporangia. That's what Rhizopus forms. Right, here's Rhizopus. It forms sporangia. Okay. Right, and that's a distinct structure. There's another one called macrocnidia, also microcnidia. These we don't see, so you won't have to recognize these, but they do exist. And so that's another type of reproductive structure. Okay. All right, any questions about that? Okay, so make sure you can recognize a sporangia. Make sure you can recognize a cnidia. Okay. So let me describe the three molds to you briefly. Penicillium. So a lot of you are familiar with this. This is a common household mold. It has that greenish color. It's a frequent food contaminant. Right? You can find it, for example, on citrus, right, like on the skin. In fact, there are some slides that we're not going to look at, but they actually just took the fungus from orange peels and used that as their sample. Okay. This one produces cnidia in chains that have these finger-like projections. Now, here's an example of what penicillium looks like. So these are cnidia. This is what we mean by the finger-like projections. So you see how it forms. I look at this as like a skeleton hand, all right? It forms those finger-like projections, right? So that's what the cnidia are. The structure below it's called sterosmata. That's what the cnidia spores form on. Okay, just to get you into the language, right? And as we've been talking about in this course, penicillium is the source of penicillin, which is a well-known antibiotic. Aspergillus is the second one. This is again a common food contaminant. It's also used as a way to make some food products. Okay. However, there are some species that are toxic. They produce mycotoxins. So one example of a mycotoxin is aflatoxin. Some of you may know where you find aflatoxin. Does anyone know? No. So there is a food item where you can find this mold, where you can find this toxin.
Is it peanuts?
Peanuts, yes. Peanuts, you can find aflatoxin. And that comes from the mold that's a common contaminant. Okay? All right, this is an example of an opportunistic pathogen. So remember, an opportunistic pathogen is an organism that usually doesn't make you sick, but it can make you sick if you are, like if your immune system is impaired. Okay? So for most of us, Aspergillus is not going to really do very much. However, in a compromised host, it can actually invade the respiratory tract. And so you can end up finding that network, that fuzz inside your lungs, which is not good, right? And again, this produces cnidia, right? Cnidia. So let's go back to looking at this slide. This shows you the penicillium, which is above. It shows you Aspergillus, which is below. Okay? They both make cnidia. So here's the cnidia spores. Here's the cnidia spores for the Aspergillus. However, one thing you'll notice is that they're arranged a lot differently. Here you get these finger-like projections. Okay. Here, what happens is that instead of getting these projections in like one localized area, like your hand, from that structure, in this case they form all around. They form a ball. Okay. So you see the cnidia form all the way around the centerpiece, called a vesicle, whereas here they only form this in a particular area, right? So that's a dead giveaway about which one is which. Okay? All right, so this one's penicillium, this one is Aspergillus. Okay? All right, any questions about that? And you can sort of see that, not as well, but you can sort of see it in this drawing, right? How they show the finger-like projections here. Okay? Whereas here they show the vesicle and they show this wrapping all the way around, right? That center vesicle. Okay? So that's what you're going to be looking for. All right, the last one is rhizopus. Rhizopus causes, again, is an opportunistic pathogen. Now, for most of the time, these really don't do anything. I mean, they don't really cause too many health problems. But this can be a wound. This can cause wound and respiratory infections in a compromised host. Okay. And this one produces a different type of reproductive structure. It's called a sporangia. Right? And the spores that make up the sporangia, which are called sporangiospores. And what that looks like is shown here. So here is a sporangia. Okay, it's a sporangia. Now, some of you, I know some people always think, well, that looks kind of like this, right? What's different though is that this is inside a sac, right? So it's actually enclosed in a sac. So there are spores, but they're within a sac, whereas these are just hanging out, right? They're actually hanging out. There's no sac around them. Okay. So these are on the outside. These are on the inside of that sac. Now, what happens is that the sporangia break open, the spores start to germinate to form the mycelia. Okay, and here we go. So what happens is that besides this asexual form of reproduction, there's also a sexual mode of reproduction. So this is the one example where you can have a sexual mode of reproduction. What that involves is mating between two different types. Okay? So here we just designate it as plus and minus. So if there's two different mating types, they can come together like this, exchange or combine genetic information to form a zygospore. Okay, so remember that this asexually, this has one set of, let's say, chromosomes. If it mates, it has how many sets? Two, right? It has two sets, right? Plus and minus. Okay? That's going to form a different type of reproductive structure called a zygospore. Okay? Or here they say zygosporangia, we call it zygospore. Right? So this is a structure that contains both sets of genetic information. Now remember that in biology, this has an important benefit. It allows for the organism to be able to diversify because now it has two sets of genetic material to work with and recombine, mix and match, right? To produce an improved variety of this organism. Okay. What happens after that is that this zygospore can germinate, and now it forms a regular sporangia, which can produce more spores, and those spores can go through this cycle. And so you have this divergence where these spores can go, if they can find their partner, they can reproduce sexually, or they can reproduce asexually. Okay, so it does both. All right. Any questions about that? Okay, so I'm going to do that. Okay, so now let me give you a breakdown of what we're going to do today. And so let me look at the lab. So one thing you're going to fill out today is, if you look at the lab, the lab has two parts. There's one part where you're going to draw what the molds look like. And you can't do that today because you have to grow the molds. So the first page you're not going to do today. The second page of the lab exercise we are going to do, which is we're going to draw the mold structures, right, which are from the slides. Okay? So the two parts of this lab, one is I'm going to show you how to culture the molds. The second part is we're going to look at the slides so you can see firsthand what these structures look like. So now let me first describe the slides to you because there's there's actually a lot of slides in there that aren't useful to us, but they're there. So I want to make sure you don't look at those slides, right? And also I want to make sure that you know what structures you're going to be looking for. Okay? So with Aspergillus, what you want to look for, the structure that you want to look for is the conidia. All right, so you want to find the conidia. So, and make sure you do a drawing, right? All right, so you want to draw the conidia. So let's say find and draw conidia. And we have the slides over here, so let me take a look to make sure I point to the right slide. So what it should say, that was exciting. Okay, so you want to use, what just came off? Nothing? Okay. I thought it was a microscope, that's all right. All right, so you want to use the Aspergillus conidiophores. That's the slide you want to use. All right, there's